Hello, welcome to the International Statistical Genetics Workshop. I'm Ben Neal, one of the course directors. And although we are crestfallen not to be in Boulder, where we typically are to see and work with all of the students in person to learn about how to pursue genetics, we've decided to take the workshop virtual. And in doing so, this virtual format, I think, offers a lot of opportunities. So these pre-recorded lectures touching on the theoretical underpinnings of the work that we're going to be describing today is definitely a bonus and an advantage. We're really, really sad not to be able to get to know all of you students in the same way. Nevertheless, we've put a lot of effort into the curriculum and into the format to provide both these theoretical lectures up front and then in-person practical tutorials over a 2-week period in June. The curriculum will include most of the major activities in genetics that are operating today as it relates to complex traits. So we'll learn about genotyping and sequencing, heritability analyses, polygenic risk scores, some biological interpretation and pathway analysis, as well as testing common and rare genetic variation for association to trait variation in the population. The curriculum itself is structured as 10 distinct days with some theory up front and some practical application in real time. All the theory lectures are available online like this one. Okay, so to dive in, I think it's important whenever talking about genetics to not necessarily start purely with genetics, but to start with this paper from 1985 from Jeffrey Rose that George Davey Smith introduced me to. And in this paper, I mean, George describes it as, if anything, attempt at crafting a theoretical basis for epidemiology as a discipline. And in a lot of ways, what we're doing in genetics, and particularly genetic epidemiology, is trying to understand different distributions of diseases or traits or risk factors in the population and how they relate to people getting sick or not sick, and how we might deal with overall major public health burdens. And so in 85, Rose showed this picture, which is the distribution of systolic blood pressure in London civil servants, a cohort recruited from London civil servants, and a cohort recruited from nomadic people in Kenya. And what you can see is that these distributions are both pretty normal, but there's a really dramatic mean difference between these groups. There's a huge, huge, huge mean difference between the blood pressure of nomadic people from Kenya and civil servants from London. Studying only the London civil servants will not teach us about this distributional difference between these populations. And I think that that's the distinction between studying individuals and studying populations, is that when you're trying to understand why someone got sick, there is a temptation to study just a single individual, or at least to try and figure out what the course of events that gave rise to this individual's presentation of a given disease is. And that's not the same as trying to understand the dynamics of why people are getting sick in that population. Another example in this paper talks about water hardness levels. And since water hardness levels are strongly correlated locally, so if everyone's pulling from the same well, they're all pulling on the same water hardness level, or there's that kind of regional variation in composition of minerals in the soil that's going to influence water hardness, that makes it extremely difficult to assess the impact of water hardness on various health-related outcomes. And so the lack of variability in an environmental exposure can be a major aspect of risk for a given disease, but it will not always be easily testable and evaluatable if you're only looking at one individual population. And in that way, genetics is very much the same. It is only looking at variation within the population. Genetics is not really going to help us unravel the difference in these distributions from the picture that Rose showed us. Indeed, most genetic analyses seem to suggest that genetic effects, such that they are, if they are present in one environment, have similar kinds of consequences more generally. But you can think about heritability and genetic analysis as trying to characterize what's going on within this population. It will not always give us this kind of between population view, and it will not give us the best look at the environmental influences on the outcome in question as well. Nevertheless, genetics is an important component of the variation within the population. And so there's a huge amount of heritability that's been estimated for a very large number of traits. I'll talk a little bit about what heritability means in a little bit later in this lecture. But fundamentally, we as geneticists are often focused on trying to understand the consequences of genetic variation and use that as a way to learn things about biology, but also potentially learn things about epidemiology through approaches like Mendelian randomization that Dave Evans will teach about a little bit later on during this course. This slide is in here to remind everyone that it is not just genotype that matters in shaping trait variation. And indeed, the environment has a massive influence, and the environment may even play a role in shaping which genetic factors do matter or don't matter, depending on what environmental conditions are available or operating at the time. If you think about the introduction of cigarette tax legislation and those kinds of things, that is an environmental change that may have an influence on the architecture, the genetic architecture of some of the traits that are a consequence of smoking. And we won't be able to necessarily decipher that just based on the genetic analysis alone. Okay, so into genetics, let's try and break it all the way down and really make sure that everyone is absolutely crystal clear on the basic principles and theoretical basis for this work. And as with many things in genetics, it goes back to Mendel, Gregor Mendel, operating in the mid-1800s, and the P experiments. This is a very, very nice picture from Wikipedia that is showing Mendel's laws of segregation and independent assortment. So we remember have a mother and a father, and we're getting half our genetic material from our mom and half our genetic material from our dad. And what Mendel did was went and collected a number of characteristics of pea plants to try and investigate which were being inherited, whether there was actually transmission from parent to offspring, and how does this transmission work? And so there are a couple of traits laid out here. The first is the color trait of green peas versus yellow peas. And the second is sort of wrinkly peas versus smooth peas. And what Mendel figured out was that green, the kind of green pigment was dominant acting. So that is to say that you would, if you had one copy of the green allele or two copies of the green allele, you would present as a green pea. And you can see that in that 3/4 of the peas are green on this slide and 1/4 are yellow. And then on the other side, there's this wrinkly versus smooth. And then again, this wrinkly is dominant and the smooth pea shell is recessive. And you can see the same 3/4 and 1/4 breakdown of the dominant versus the recessive, because the genotypes can break down in that kind of way. Hopefully this is stuff that you all learned in like the basic biology, introduction to genetic stuff and all that kind of stuff. So I'm not going to belabor it too much here, but these principles of segregation and independent assortment for discrete characteristics like color or wrinkly or smooth were worked out and then really popularized at the turn of the 20th century by a guy named Bateson. However, even in Mendelian genetics, there's a little bit of nuance introduced, particularly as it related to something like codominance. And so here, rather than there being a dominant and recessive, there was this codominance idea where the parental generation of this flower breeding experiment has white flowers and red flowers. You cross those, you get this F1 generation, the first offspring generation, and you get pink. You get nothing but pink flowers crossing red flowers and white flowers in that offspring generation. If you go and do a so-called back cross, so take the pink flower and cross it with a white flower, you end up with the 50-50 pink and white. So the kind of one red allele is coming from the red-white heterozygous, pink flower. And that ends up with a pink flower in the subsequent generation. And then that's in contrast to this, other half the time where you get just the white flower because the white allele is being homozygosed. if you inbreed within this F1 generation, the pink flowers, you end up recapitulating a quarter, half a quarter. So again, with this notion that half the genetic material is coming from your mom and half your genetic material is coming from your dad. This was understood as a phenomenon before the genome was appreciated, before we understood what nucleic acids were. I mean, before we even really had any sort of sophisticated view of cell biology in any sort of meaningful way, certainly as it relates to genetics and inheritance. And that's a real kind of striking thing to think about, is that just based on observing these patterns, trying to collect some data and do some experiments and testing your hypotheses, you can learn these rules of biology. And that, I think, is really important and exciting. The challenge, however, with the sort of purely Mendelian view of the world is that Mendelian characteristics were a really beautiful explanation of a complicated problem. Why are some peas certain colors, or why are they wrinkly or smooth? But it didn't deal with another observation. So this is a guy over here on the left, a kind of Victorian scientist, very severe looking gentleman, is Francis Galton. Galton is a complex figure in the history of genetics. This picture here, I'll talk about the high points before I get to the low points. This picture here shows the distribution of parental height. So that's this A to B line here. That's the average height of parents. And then plotted in the same position on this x-axis as the mid-parental height is the offspring height. And you can see this C to D line here. That's a different line drawn. And what you can see is that there is familial resemblance, that parents that are shorter than average tend to have shorter than average kids, although not quite as short. And likewise, parents that are taller than average over here tend to have taller than average kids, although not quite as tall. And indeed, that kind of movement closer to the population mean is where Galton coined the term regression to describe drawing the line through the data points. So he's actually describing this C to D line that he's drawn here as the regression line of the kid height, and it's actually regressing to the mean away from the deviation that's observed in the parental generation. And so here we have this continuous characteristic, height, okay? This is how tall you are. But how do you square that with the Mendelian characteristic view? How do you make sense of this as a coherent way? Why are we similar on something like height that doesn't have, like, there isn't a I'm 6'4" and therefore I have a single Mendelian characteristic that I've inherited that makes me 6'4"? Where does this come from? And this was a problem that actually gripped the scientific literature throughout the late 19th and early 20th century. There was a huge amount of debate about that. And before I get to the solving the riddle part, I do just want to take a moment to flag that Galton, like a number of the other scientists that I'm going to talk about today, were eugenicists. And actually, Galton has the distinction of coining the term eugenics, which is got a very difficult and complex history associated with lots of really nefarious things, particularly around racism, around immigration laws, around sterilization, a lot of these things that are really traumatic. There was a lot of pseudoscience in eugenics as well, and we think it's so important that we have a lecture dedicated to the societal aspects and dimensions of this that Leah Davis is going to give, and I encourage you to watch that as well, because I think it's a really important part of what our responsibilities as scientists are in terms of making sure that we achieve beneficence. That is, that there are positive outcomes, that there is an upside, there's a clear benefit to humanity for the work that we do. Okay, so back to the science narrative. So we got Galton, continuous variations, similar in families, something like height. How do we make sense of this? There's got to be some kind of genetic stuff, but what does that mean? And there's a really nice piece of work by East in 1916 looking at the inheritance of Corolla length. And I just grabbed an early paragraph from that paper because I think it both illustrates many of the debates that we're having today about genetics have not changed in a century in some ways, that there are still elements of this dialogue going on in our discipline, what it means, how it works, and how it operates. But it was really this striking difference in our ability to provide a simple and coherent and sufficient explanation for a characteristic like P color with continuous trait. Now, in this instance, it's not height, it's actually the corolla length in the tobacco plant. Corolla is just a fancy science jargon word for all the petals of a flower. So the kind of overall flower petal length might be a better way of describing this, at least a little more accessible, but nevertheless. And so what East did was he took a set of tobacco plants that were self-fertilizing and isolated the extremes of the distribution of corolla length. So he took the very short corolla length over here and the very tall corolla length over there and made that the parental generation, and then crossed it with its, across those two to create this F1 generation, right? So we see this first offspring generation, F1, and its mean is pretty close to the average mean of the parental generation. So the mean's in about the same place. And so that means that somehow, some way or other, these plants, such as they are, have things that, even though it's very short over here and very tall over here, it ends up in the midpoint in the F1 generation. So that's kind of interesting. Even more interesting is that it's not just as if these are like a small set of fixed characteristics. It's that if you cross that F1 generation within itself to get an F2 generation, you see an expansion of the variability in that distribution. That expansion in the variability of the distribution in corolla length is really, really important, because what it suggested was that the long corolla length had length increasing alleles, homozygous, so 2 copies of it, like as if there's no more variation, and then 2 short corona length alleles, homozygous. And so there's very little variation over in that bottom hand distribution. And so you now introduce a bunch of pure heterozygotes, or instances where you have one allele and the other allele, and I'll get to more formal definitions of those in a moment, at the kind of across the entire genome. And now you're going to get a reshuffling of those with the fertilization within the F1 generation. So F2 generation should have much more variability than F1 generation if some of these heterozygocytes now have an opportunity to go back to their homozygous long or homozygous short versions that might have been homozygous in the parental generation. Further supporting this was an observation that you can take from any point in this F2 generation, breed within that point, and then you'll end up with a mean, a population mean value that's pretty close to where that point is, and you will recapitulate pretty much all of the variability that you see in that F2 generation. And that's really a tremendous set of plant experiments to demonstrate that polygenic inheritance is likely to be the case. Now, again, no idea about the genome, no structure of DNA known. This was well before nucleic acids were really worked out as what was living in the middle of cells, like it's starting to happen at this point in time in the kind of scientific tradition. So huge, huge, huge inductive reasoning work to figure out this polygenic model. Now, this is a good set of experiments to demonstrate that polygenicity may be operating, but it is not a kind of formal model for expressing precisely what is going on. And for that, we really have Ronald Fisher to thank. So there's a picture of Ronald Fisher from 1913 from Wikipedia. Fisher, another eugenicist, another very complex and controversial figure, some pretty heinous views on class and society that obviously he was taught, but he didn't reject. So there's that. But nevertheless, a brilliant scientist as well. Truly, truly, truly understood things about how to kind of do science that has transformed the way that many do science writ large. This Design of Experiments book really lays out a lot of what we think about when we're trying to do statistical research. I mean, this is a foundational, if not the foundational work in modern statistics. And so I kind of highly recommend that if you want to understand Fisherian statistics well. But for the purposes of this lecture, I'm going to focus my attention on the Fisher 1918 truly landmark work of genius, the correlation between relatives on the supposition of Mendelian inheritance. In this paper, Fisher developed a number of things that we use today still very regularly, including variance. So like the formal definition of variance as we use it now was here, although Gauss, and if you want another genius who really understood a tremendous amount about how to express the world in terms of math, Gauss is really the one to go take a look at. But nevertheless, Fisher defined variance. He also tried to create a framework to describe this polygenic inheritance. How do we reconcile this continuous variation in height and the similarity amongst siblings in height and the similarity amongst parent and offspring in height with the discrete inherited Mendelian characteristic work that was so clearly laid out by Mendel and then popularized by Bateson? Good question. So how does Fisher approach this? Well, he kind of combined a couple of things. The first and most important thing is a quick crash course on something called the central limit theorem. And basically what the central limit theorem says, that if we imagine each genotype here as a coin toss of having one version or another version of an allele, if we only have one coin and we count the number of, say, heads minus tails as the kind of outcome, or the number of heads and the number of tails as the outcome, however you want to design the outcome, you get this distribution that's kind of 50-50 equally split, assuming your coin is fair. If we add some more coin tosses to this, well, we now get more different outcomes. Okay, so we get, two heads, heads, tails, tails, heads, or tails, tails. And so if we ignore order, which for the sake of the genome makes sense because there's no particular reason why a height increasing allele on chromosome 1 should be fundamentally different than a height increasing allele on chromosome 3 necessarily. So we, get slightly more variability in our distribution. And then something interesting happens when we keep adding coins. So as we keep adding coins, we start to see a distribution emerge. And it's a distribution that you should all be pretty familiar with, is this normal distribution. And so what the central limit theorem states is that if you sum a large number of independent factors that are identically distributed, then you will get a emergent normal distribution. And what it means is that if you add a bunch of stuff up that are all approximately equal weight, give or take, you have some variability in that, you'll end up with a largely normal distribution. Gauss characterized this in 1827. We call it a Gaussian, so famous was his description of it, but really the original description of the normal distribution goes back to de Moivre in 1738, at least that we can track down in the tradition of sort of Western mathematical development. This may have been discovered many other places at many other points in time. So in any event, we have this normal distribution. So the central limit theorem is one important piece of this theory, that we've got lots of independent things that we're summing up. And then what are those things that we're summing up? Well, that's a good question, but just a quick sidebar for the liability threshold model. So you might ask for something like diabetes or schizophrenia. That's not a normal distribution. There isn't a normal distribution in the population. And that's true. There isn't this normal distribution of diabetes or not in the population. It's really binary. And so it's got a distribution that looks a little bit more like that coin toss, but weighted towards the prevalence of diabetes. In 1900, Carl Pearson and Alice Lee, and actually I encourage you to read this one as well. You can kind of click this link and follow to the work itself. Pearson writes a really nice introduction about how he couldn't have done this without Alice Lee. And so even then there were complexities around representation of women in science, and that it was not uniformly held that women should have positions and roles in science. And it was pretty clear that Pearson didn't necessarily totally agree with that. And he certainly wanted to ensure and recognize the contribution of Alice Lee to a lot of the foundational theory around how we think about discrete characteristics as they relate to normal distributions. And so the observation that Pearson and Lee made in 1900 was that you could imagine lining up every single horse from the kind of whitest white to the blackest black in terms of horse coat color, and that might create underlying distribution of horse coat color, of pigmentation in horse coat color, and that could be ordered, and that single distribution, you could imagine chopping it up at different places and have different colors of horses at different places chopped up in the distribution, still allowing for this normal distribution. And so even for a binary outcome, a kind of reasonable model may well be to take this kind of extreme tail of the distribution and assume that there's an underlying distribution of liability that gives rise to this particular outcome. This liability threshold model is hugely, hugely important. I think Falconer probably popularized it most aggressively in the 60s, and he sometimes even gets credited for inventing it. But as with many things in science, there's just this process of rediscovery time and time again of some set of ideas. Okay, so back to that liability distribution, or that normal distribution that is describing height. So we have the central limit theorem to get this emergent normal distribution. But what are these Mendelian characteristics that Fisher is adding up together? Well, this is a lovely set of slides that Manuel Ferreira, a former tutor on this workshop, put together, that I'm shamelessly reusing and providing appropriate citation for. And what, Fisher noted with the Mendelian characteristics was he was like, let's just imagine for a moment that we don't have just one genetic effect, but we have lots of genetic effects, and that these genetic effects have some influence on trait value. Okay, so something like height. Now, the simplest form of this in Fisher's mind was to take a purely additive model. So that is to say, this sort of little a, so this is one of the two alleles at the genotype position that's having an influence on the outcome. And Fisher did this, again, without structure of the genome, like none of that. It's really, really impressive. Genetic variation was a hypothesized, like a kind of assumed phenomenon, not a known, clear, understood phenomenon in terms of what was going on. And there was a big debate about whether it was all somatic, all arising in the organism as it was living, or whether it was inherited, that this was variation that was running around the population. Big open questions. In any event, so we take these individual Mendelian characteristics, and we ask that, and we set up this model of having this additive genetic effect, this plus a. So we can see that little a to big A has a distance of plus a. Then we have big A to big A also has this distance of plus a. So this is an additive genetic effect. So this is a locus with just additive genetic variance. No dominance. The dominance deviation, the midpoint between the two homozygotes, as it was defined by Fisher, equals 0. Now, you can think of this as actually a source of variation in the trait, because remember, if you have little a at the genotype, this genotype is not capturing all of the contribution to variability that your phenotype possesses. There's no single genotype that captures your whole height. There's, under this model, this polygenic model, there are lots of independent genetic effects that might be contributing, and environmental effects, and a whole host of other things that you can kind of think about and start to write down and theorize about, and maybe even test if we can collect sufficient data to really examine and look at that. And so, but nevertheless, we have these conditional genotype means now, right? So that's what this model implies, that the mean value for the little a genotype is different than the mean value for the big A genotype, is different from the mean value of the big A genotype. Therefore, it is a source of variation. It is creating some variability on this trait in this population. And what we've got drawn here are kind of pretend genotype-centered mean distributions with residual variability. And you can see that these do create additional variance. There's more spread overall than there is in any individual genotype group. And that is the source of variation that we're trying to understand when we're talking about heritability. Now, we can move beyond this a little bit. We can introduce dominance into it. And dominance here is a situation such that the heterozygote is not smack dab in between the two homozygotes. So that is to say, big A, little a has deviated from the midpoint of little a, and big A, big A. And that deviation from that midpoint, that's the dominance deviation. And sometimes people refer to dominance or as just dominance not equaling 0. There is occasionally a circumstance referred to as overdominance, such that big A, little a as a genotype ends up outside of the homozygotes. This is observed sometimes in the plant literature and for some human traits, but by and large, there's not a huge amount of support for this overdominance construct, but nevertheless, it's all the same to the math, and that, I guess, is where Fisher came to the problem with. Okay, so here we now have a deviation from that heterozygote, and that actually has the potential to contribute variability as well. It's just not the same variability. And so you could think of this, another way of thinking about this is that each genotype has its own mean that do not have stable distances between those genotypic means. That's all that it's trying to soak up. So it's the allele, the pair of alleles at the position has some consequence. Okay. So now let's go to statistics. So we're going to pivot from the genetic model to statistics, because really Fisher 1918 was not only the writing down of heritability and the writing down of the sort of ways of thinking about genetic consequences, it was also the writing down of variance. And I mentioned this already, but variance is really, really, really important because much of statistics is dedicated to understanding sources of variability. So first, just start with the mean, nice and simple. The mean is the average value of all the traits. You're all introduced to this in your basic stats, I'm sure. But the variance, this variance property, this was introduced in this 1918 paper. And the reason that Fisher picked the sum of the squared deviation from the mean as the estimator of the spread of the distribution was that this estimator of variance has the least variance. So he used his own statistics to figure out what statistics to use. This is really fiendishly clever, but it makes sense. You want your statistic to be as unvariable as possible, to have as little spread as possible. And so the whole introduction of the variance was around the idea that this is going to give you the best possible estimator for characterizing the spread and the distribution overall. Okay, we don't just have variances in life because we don't just have one trait, we have multiple traits. And so when thinking about more than one trait, you can think about both the individual means, so the mean of X, so X is trait 1 and Y is trait 2. And you can think about X sub I as the individual observations of the X trait in individual I, and Y sub I as the observations of the Y'th trait in individual I. And so now we have the deviations from the mean of X's times the deviation from the mean in Y's. And that is now a representation of the covariation or the covariance. And covariance is really powerful as well because it is a way to measure the amount of sharing a pair of traits has in some way. I mean, you can standardize this, dividing it by the variances, and that'll get you to the correlation. Okay, so those are means and variances and covariances. Go grub around on Wikipedia or look for Intro to Stats 101 if you want more background on that. I'm going to return us to genetics and to the kind of statistical genetic theory that was introduced by Fisher in 1918. And so we've got, again, our mean here. We can define the mean as the sum of X sub I. So that's the individual observations of the trait X in individual I, times the frequency of X sub I. So if you just think about counting how many times you observe a given value and then have that as the kind of overall frequency, divide that by the total number of observations, that'll give you an estimate of the mean when you multiply those values by the value that you observe itself, just calculating the mean. Okay, so we take the mean as the assumption. And I introduced this idea that the genotype can be a source of variation in the phenotype, that those different genotypic means creates variability in the overall distribution. Well, we can think about this as a concrete quantitative readout, something like cholesterol levels in the population. we need to know what our genotypes are. We need to know what the effect of having the genotype is. And we need to know the frequencies of those genotypes to get to the overall grand mean and to get to what the contribution to phenotypic variance this particular genetic effect is. Okay, so the mean we might define here is this. So we've got the A is the effect. Remember, this is sort of the value of the genotype conditional mean for genotype AA. So that's the trait value. And that will occur with p squared times. So I've introduced here subtly P and Q is notation for the frequency of big A and little a alleles. So p squared is big A. So that's having one, having a big A from your mom and a big A from your dad means that you should see that approximately p squared amount of times in the population, versus 2pq, where you can have a big A from your mom and a little a from your dad, or a big A from your dad and a little a from your mom. And that'll happen 2pq because the order doesn't matter whether you got it from your mom or dad. And I'm not going to talk about all of the other exceptions to that, but by and large, just for simplest case, let's just assume that that's true. And then little a is q squared. So that's, you got a little a from your mom and a little a from your dad. So those are the, in a sense, frequencies, the allele frequencies of the genotype frequencies of these organizations of genotypes under an assumption of Hardy-Weinberg. And the mean in the overall population is the trait value times the frequency summed for all trait values and frequencies, as we described a little bit earlier. And that you can turn through a little bit of algebra into A times P minus Q plus 2PQ times D. Okay, so this is the trait mean. This is the grand mean in the population. But now we got to do the same thing with variance. So recall, for the variance, we're no longer just interested in the individual observations and its deviation from the mean, but we're going to square those and we're going to sum those squares, again, weighted by the frequency of those contributions. So we're going to work through the same exact math here. So we've got our same genotypes broken down, our same frequencies. All of that is exactly the same as the mean case. But to determine what the variance is, well, we have to calculate what the x sub I minus mu, or the individual observation by the mean quantity squared is. So here we know that the trait value is A, and we have the mean from before that we worked out, m. So that's A minus m squared times p squared, plus d minus m squared times 2pq, plus minus A minus m squared times q squared. If you do some algebra and do some rearrangement, you'll find that this turns into the variance of the QTL. It's also reduced form is 2pq quantity A plus q minus p d quantity squared. And so that now gives us a way to estimate how much of the variance. Okay, so here we'll go through this derivation line by line. So starting with the variance of x. So this is the overall variability that we saw from before. Here's the reduction that I alluded to, this 2pq A plus q minus pd quantity squared. That's the additive only part. And then we also get this other component here, this 2pq d quantity squared. That's the dominance component. So this is the partitioning of the additive and dominance contribution to trait variation that Fisher introduced. Now, this decomposition is the subject to a fair amount of scientific work because indeed, the additive model has a fair amount of robustness across a variety of values of D. So the additive only model sometimes gets preferenced, not because it is necessarily exactly how the gene action is happening biologically, but more because it does a very good job of absorbing most of the variability in the system. Okay, so another way of saying this is in words rather than in math, which is the additive effects are the main effect. That's just a 1 degree of freedom test on your genotypes against your trait value. And then the dominance effects are the deviation from that additive effect. So it really is almost, in a sense, a second order effect after the first order effect of the kind of genotypic means, which is why the additivity soaks up a lot of variability. And indeed, in the additive variance, you can see right here, right in here, that there's the dominance term. So the dominance deviation comes out as a contribution to additive genetic variance, unless Q minus P equals 0. So a 50-50 allele will never see any dominance deviation coming out in the additive genetic variance. Or you're in this kind of weird circumstance where A and Q minus PD cancel. Okay, that's a pretty small space of potential models. You can contrast that to this thing over here, 2pq d². Remember, p and q are less than 1, and as a result, as long as if d is less than 1, then it is certainly the case that this will always be less than this. If d is greater than 1, it gets a little squirrely, but by and large, the additive genetic variance is going to be the lion's share of the genetic variance just based on the way the math is designed. And it is as simple as that. And I know that's not a very satisfying explanation, but that is the way it works out. Okay, now continuing on. So that's one, but remember, we have polygenes. So the whole Fisherian idea was to bring that idea of single genetic variant action together with the central limit theorem to get at that polygenic model. And those many small genetic effects, there was lots of experiments in the plant and animal literature about kind of empirical demonstrations of things that would be consistent with polygenicity are what we observe in the experimental work. Now we can develop these ideas a little bit further and maybe impose some distribution on genetic effects. So if we say that the distribution of genetic effects has a mean centered at 0 and it has some variance associated with it that maybe we can estimate, well, if we assume those distributional properties, we can actually now start to get at estimation of heritability. And so this was the real kind of special sauce underneath GCTA that you'll hear a lot about this coming 2 weeks, where we take this polygenic inheritance model, this concept of familial resemblance being then turned into a concrete genetic variation having an impact on phenotypic variation, and then summing up those contributions over the genome to get at this estimate of heritability. We can then go one step further and kind of try and build models about how those genetic effects might be distributed and use those models to get at estimates of the overall contribution of genetic variance to phenotypic variance. We also did this not based on the genetic variation itself, which is what is in GCTA, but looking at genetic association summary statistics. And I'll come to summary statistics a little bit further along in this lecture. Okay. Fisher 1918 has a ton of conceptual work in it, many of which we're still figuring out and resolving and testing and trying to explore and understand today. Included in that are not only what the expected correlations are amongst parent and offspring and amongst siblings or fraternal correlations or correlations amongst other relatives and what is implied therein, but also other phenomena about how genetic variation might be influencing trait variation. Things like assortative mating. So if random mating is not operating in the population, what does that do to this kind of model? Or how this generalizes to variation with more than 2 alleles. If there are many, many, many more alleles, what happens with that? Or various things around the kind of relationship with kind of dominance deviation and how to detect and think about all of those kinds of things. And I think the thing that's really nice about this work is that many of the ideas that are in it are still relevant today and are still relevant to the work that we do today. And so even though it's challenging to get through, do have a go. George Davies Smith also did everyone a favor by putting an updated corrected version in the International Journal of Epidemiology, and we'll make that available to all the students as well. But there are some really interesting ideas in this, in some of the early work from East, in some of the, this scientific literature is becoming more accessible, not less accessible with the rise of the internet. And so I would really encourage you, if you're interested, to go out and read the primary evidence for yourself and try and learn that way, because it'll give you a completely different appreciation for a lot of the debates and discussions about genetics today are all laid out in a tremendous amount of detail over 100 years ago. Okay, so that's like the statistical genetic theory. That's the theory of variance. Variance is really important in statistics, not just statistical genetics. But what about the genome? What about genetic variation? I've been making allusions to all of these things all the way through. I think it's important to just document in detail exactly what I mean when I'm talking about some of these different phenomena for pedagogical purposes. So structure of the genome, 1953, Watson and Crick, really another landmark foundational piece of work. Go read about Rosalind Franklin. If you don't know who Rosalind Franklin is, go read about her. Strongly recommend that. Watson is another one of these characters in science that has some very problematic positions in terms of eugenics and the things that he's, kind of supportive of and all of those sorts of things. But nevertheless, the scientific work is also important because it teaches us something about the world around us and describes something truthfully. And describing something truthfully is what we hope for in science, or at least that's what I hope for in science. But just as a reminder, we've got 4 DNA bases, A, C, G, and T. That double helix is the structure, and the bases are paired such that A goes with T and C goes with G, with hydrogen bonds to keep the, in a sense, zipper zipped up. There are a couple of different structures of the genome. It organizes itself in a couple of different ways. So there are different couple of flavors of DNA out there that you can read about. But by and large, this is the material of life, some would style it. And that's what is in basically all of our cells in our body. And it has the instructions for our growth and development, as well as the maintenance of all the things that we do on a day-to-day basis. Now, the genome itself has our human genomes have 23 pairs of chromosomes, 1 from the mom, 1 from the dad. There are about 20,000 genes in the genome. This picture over here on the left is called a karyotype or karyogram. And what that is it's basically isolating DNA from a cell that's going through mitosis and then splatting it against a slide and then arranging it based on size. And actually the numbering of chromosomes comes from the ordering of them by size. And that's where the numbers come from. It's not perfect. I think that chromosome 20 is the shortest chromosome, maybe 21 is the shortest chromosome, not 22. So there's a little bit of fiddliness in the back end of the chromosomal distribution, but approximately it tracks the size pretty well. 20,000 genes, give or take. Genes get transcribed. So DNA gets transcribed into RNA. So the coding regions of the genome gets transcribed into these transcripts. And then those transcripts, that mRNA, that messenger RNA, gets translated into proteins. And then that protein is comprised of an amino acid sequence. So it's a bunch of amino acids sort of glued together end to end as the way proteins are made. Proteins make almost all of you. And so, this is the stuff that life is made of, as they say. Now, that's just the genome. The first mapping of the human genome, or at least the first draft sequence of the map of the human genome, all 3 billion plus bases of it, was published in 2001. This was a large-scale international collaborative effort that changed the way geneticists did science. Prior to the Human Genome Project, I think genetics and genomics was much more competitive than it is today, where collaboration and consortium work are more the rule than the exception, I would almost go so far as to say. And a lot of that mindset changed with trying to scale the mountain of a task that was the human genome. This was a scientific project heretofore never seen in biology. It was probably more akin to something like the Manhattan Project than anything else. And it was really just a start. I mean, Eric Lander was asked to describe the human genome in a few words. And I think he said something to the effect of, the genome bought the book hard to read. And he was exactly right. It's extremely difficult to read the human genome, and it's still very difficult to read the human genome in terms of what it's actually doing right now. But just mapping the human genome, that was really just phase one. There's genetic variation all in the human population. There's all of this genetic variation is contributing to all the phenotypic variation that we're interested in studying. And that phenotypic, that genetic variation was really collected and aggregated through efforts like the International HapMap Project, or its forerunner, something called the SNP Consortium. This also gave rise to genome-wide association technology, also called common variant association technology or common variant association studies, or the technology itself is called SNP chips or SNP arrays or genotyping arrays, some blend of those words. And the way they work is they've got a bunch of bases of nucleic acids all lined up, and they are there to match specific sequences drawn from the human genome with a goal to detecting what version of the particular genetic variant you have at that position. So these arrays really came about as a consequence of having large-scale surveys of common genetic variation available. And then more recently, we've seen the emergence of next-generation sequencing. I'll talk a little bit about those different technologies a bit later on. But sequencing enables you to capture a much larger class of genetic variation. So the SNP chips only allow you to test the things that you know about already, in a sense, whereas sequencing allows for genetic variant discovery as well as genotyping of known genetic variants, and as a result is really powerful for the investigation of rare genetic variation in contrast to common genetic variation. We will teach about all of these kinds of genetic variation over the course of this workshop. Okay, so onto the technology. So we've got arrays, and then there are sort of two main flavors and approaches to use for exome sequencing versus genome sequencing. So exome sequencing is targeting just the coding region of the genome, so just the genes, about a little over 1% of the genome, so much smaller target. And that means that you can do a lot more people, which is really nice. Whereas genomes just try and go genome-wide. They chop up the genomes and then sequence. It is worth remembering that the current technologies are primarily short read sequence. So that means you get maybe somewhere in the neighborhood of 150 to 300 bases of sequence per little fragment, and then you got to go align it and then figure out whether or not there's a genetic variant there and all that kind of stuff. But read length is something to be cognizant of when thinking about sequencing technologies, since there are other longer read sequencing technologies that are out there that have different upsides and downsides as well. Okay, so the main upsides for arrays are that they've been around longest. They are the most widely used in human genetic inquiry right now. This is the GWAS engine. The 100,000 plus common variant associations that you might have heard of have been derived from primarily working with genotyping arrays. And there's a huge amount of heritability trapped in common genetic variation. And so not only are arrays good for locus discovery, finding new places in the genome where a genetic variant is likely to have some consequence that relates to the phenotype that you're trying to study, but also for epidemiological purposes. So heritability and more variants explained means more power in epidemiological analyses. And so there are real reasons to make sure that common genetic variation still stays in view, even though rare variation is now identifiable and measurable through exome or genome sequencing technologies. Exomes sort of strike a balance. So it's a focused view of the genome, just a small subset of the genome, but it is by far the most interpretable part of the genome. The coding variation is got the most immediately interpretable biological likely consequence from it of any class of variation in the human genome. And that's really important because we want to learn biology, at least for some of the things that we're doing. We definitely are very interested in learning about the biology of what's going on, because if we understand the biology better, then that lays the groundwork for potentially improving how we think about identifying people at risk, how we think about potentially treating people, what mechanisms of action for treatment might be. All of that work can flow from a better understanding of the biological basis of human health and disease. And so exomes give you the most tractable class of genetic variation to study that is most readily and immediately interpretable. Further, there are genomes. So genomes get everything: common, rare, coding, non-coding, genome-wide, goes across the whole thing. And that's really great. But that also gives rise to what the most natural downside is, which is smaller n. So genome sequencing is still more expensive than exome sequencing. Both sequencing technologies are typically more expensive than genotyping technologies. But there are still downsides with each of these technologies, right? This is a choice. It's not like it's a clear choice. It's a balancing act between different objectives. So common genetic variation stands the problem of interpreting the hits. Danielle Posthumus' session will be devoted to the biological interpretation of genetic results. And that is something that we still struggle with. We do not understand what genetic variants do terribly well. And that seems problematic since we're geneticists and we're meant to be figuring out what genetic variants do. Exomes are good for gene identifications that can jumpstart biological inquiry, immediately prompt novel hypotheses, all of those things. But it often comes with a limited scope. We don't have a comprehensive capture of all genetic variation. That's, narrow. And being systematic and going genome wide has been a boon to our science, not a barrier. And so there's probably a shelf life on this that's related to the cost curves associated with doing genomes versus exomes and the cost curves of doing arrays versus genomes. An array plus an exome gets you a lot of the way there, right? You get the rare coding variation, the most interpretable. Rare non-coding variation is very hard to interpret and therefore very, very difficult to test. Whereas common genetic variation still has the largest tractable source of heritability that we can measure at this point in time. And so the sort of upsides of heritability can be serviced with the arrays, and then the upsides of biological inquiry can be serviced with exome sequencing. But I think it's not an unreasonable expectation that as costs continue to decline, moving to comprehensive evaluation of genetic variation through genome sequencing is where we're heading. Okay, let's talk about genetic variants for a moment, just a few classes and kinds. So I'll start with SNPs and spend most of the time talking about SNPs, or single nucleotide polymorphisms, because they're the most pervasive kind of genetic variant in the human genome. There's more of these than any other kind of variation. They're common and rare. They do all kinds of things. There are point mutations that cause you to break one copy of a gene, for example, and create a protein truncating variant. So here we have our overall DNA sequence. So the gray is where everyone matches and is all the same. And let's just say that that's the reference for the sake of argument. And then we have this position here, SNP1, where you can see the A base. So that's the DNA base that the person 1 has from their mom and their dad, as well as person 2 from their mom, whereas person 2 is the C base from their dad, or the paternal line here. And then person 3 is now homozygous for the C allele. So they've got both from the mom and the dad, the C allele at this position as SNP1. SNP1, we would say, is more common than SNP2, since there's only one copy of the T allele compared to all of the Gs, versus the 50-50 split that you see in the AC polymorphism over here. The genotypes for these two SNPs are laid out on the right-hand side, where person 1 has genotype AA at SNP1 and GG at SNP2. AC, or heterozygous genotype, at SNP1 for person 2, or GG, homozygous, for SNP2. And then person 3 is homozygous for the CC genotype and heterozygous for the GT genotype. This is a mutation that has arisen at some point in demographic history. It may have arisen multiple times, and indeed, there are likely many, many, many recurrent mutations running around the population. Typically, SNPs have two alleles. The alleles reflect the base at the position. So this would be called the A allele and the C allele for SNP1. And usually each SNP is given an RS number, although RS numbers, while useful in arrays, may be becoming deprecated in sequencing. But generally an RS number relates to a chromosome in position, sometimes, and an allele, but sometimes it's not as specific about the variant as you would hope for that particular allele. In addition to these point mutations, there are also structural variants. This is a picture that I grabbed from EBI Embl. about different kinds of genomic variation. So here down at the bottom, you see copy number variation. And so this gray block here, this gray block that is variable in the number of copies, that's referred to a copy number variant. And generally, a copy number variant is thought of as a large chunk of the genome. So, thousands of bases or more, maybe even 1 million bases that have either been deleted or duplicated. So over here in the deletion, you can see you've got this chunk of the genome. That gray bit isn't observed in the person's genome. That's a deletion. Or an insertion where you've got like a new bit of genome that isn't represented in the reference. So that's the deletion and the insertion. You can also have a duplication event. So here a chunk of the genome copies itself and inserts itself next to where it's copied itself. There's kind of mechanism around transcription that this is thought to operate through. You can go and dig into that if you're interested, but these duplications kind of expand the genome through a copying mechanism rather than the incorporation of new DNA that you see more in the insertion side. And then when you have deletions or duplications at a specific position that are variable in the population, people sometimes refer to that as copy number variation, where you have a variable number of copies of that chunk of the genome. Last up is this inversion. Inversions are really weird. They are a chunk of the genome that is taken out and then flipped around and put back in again. Okay, so everything is all turned around. And then all of the genes that are transcribed in the opposite direction to the way that they would normally be transcribed. So the gene product itself is preserved exactly. So all of the gene functions are the same. What is impeded is this process of recombination, the kind of crossing over of chromosomes that happens during meiosis so as to create the germ cells for your zygote. So, either sperm or egg cells are made via meiosis. And in meiosis, there's this process of recombination where the chromosomes basically take stretches of one another, break, and then reattach themselves, mixing up your grandmother and grandfather's genome into your parent's genome, is a kind of mix of the grandmaternal and grandpaternal genetic variation coming into you. So there's variability in that through this process of recombination. Okay, and so inversions suppress recombination because the genomes no longer line up on the other chromosomes because they're, so it's kind of like everything is upside down and backwards, but it's okay, I think is a good way of thinking about it. Okay. Another slide of definitions, coming at them again, because we will use this jargon over and over and over again. I'm really sorry about that, but jargon is really, really helpful for speeding up discussion around complex technical considerations. So here we've got SNPs. So these are just points on our idealized chromosome, the black line that is the chromosome. The haplotypes represent the entire one of the 2 copies of your genome. You can think of all of the genome that you inherited from your mom, like half of your genetic material, as one haplotype, broken up into, in a sense, 23 haplotypes. But often when we talk about haplotypes, we typically mean in a much narrower, smaller set rather than the entire paternal haplotype, so to speak, or maternal haplotype. But nevertheless, the haplotypes are what is happening on one chromosome. So it is not just simply what your genotypes are. It is what the sequence of bases are in that particular chromosome, that arm of that chromosome. It's not overall. And that's in contrast to genotypes, which take that is about a single position, and it's about what genetic variation you have at that particular position. And then the alleles are used to refer to the individual forms of the genetic variant at that position. And we're going to work with this kind of data with phenotypes, with things like disease status of affected or unaffected for something like schizophrenia or diabetes, or some trait value like height or weight or BMI or neuroticism or whatever you want. And those are the things that we're going to work with in terms of statistical genetics. We're going to try and relate genetic variation to phenotypic variation and understand what that relationship really looks like. Now, allelic association That's what Fisher was on about in the Fisher 1918 paper. He was trying to describe the mean difference that would be introduced from having an allele, and the degree to which that genetic variation yields phenotypic variation. That was the math to describe that phenomenon. So that was really the basis for those kinds of considerations. To test these things, we generally perform very, very simple regression association models. A linear model is pretty good for continuous phenotype. You can sometimes even use that for a discrete outcome, depending on what you're trying to do. I think a logistic regression model is probably more advisable, but nevertheless, it depends on what you're trying to do and the circumstances therein. And you can just code it as a simple dummy variable. This x sub I, which is the variable that is encoding the genotype information in the analysis, you can code as 1, 0 if you have a given allele. So this would be a kind of dominant model. Alternately, it could be the number of copies of the allele. So it could be like 0, 1, 2, just like in the genotypic mean example I had a bit earlier. So those are the basics to association, the basics to the genome, and the basics to population, kind of statistical genetic theory. You'll get some population genetics theory from Alex Blumendahl, and Leah Davis is going to give you some information about how to build a human genetics that works for society rather than can kind of lead us astray. We are absolutely crestfallen again that we can't be in person, but hopefully these online lectures will be useful and that you can refer to them multiple times. And we look forward to seeing you in person in the workshop in the tutor sessions. Thank you so much and take care.